some really pivotal things that were done back then. But now we're about to go into the engine room. You know, but now that we've got this thing originated, what really unfolded in the following years that actually made astrobiology what it is today. And so this is the evolution and distribution, developing the community in science. And a very key part of any community and science are the events where people get together and really discuss the science and network and all of those good things. And for that, we're going to hear from Lynn Rothschild on the birth of AbSciCon.
Well, Dave, I've been called a lot of things, particularly when I was running AbSciCon, but never the engine room. So thank you. I'll add that. Pain in the neck and various other things. Oh, I should have brought this. By the way, the first lesson I learned, this was from one of the gifts at the first AbSciCon in 2002, is we had a pen with the 3 questions in astrobiology that you click through, and I left in my bag. Maybe Rocco can rummage through. But one lesson I learned is you never have a giveaway pen at a meeting that clicks, because my whole recollection is the whole first talk was people testing the pen. So there were a lot of lessons learned. So I asked to have this not AbSciCon 2002, but the birth of AbSciCon, and there were an awful lot of people involved, and you'll see this in some of the eye charts. But my recollection is the Musketeers, which I think were also the kitchen cabinet, and I probably should have added Lynn's name on there. But really, this was Dave Morrison's brainchild. Greg Schmidt was there with me the whole way. Bruce Jakowski was, whether he wanted to or not. And then Karen Bradford, Ro Christensen, Joe Manafra, and Shirley Berthold were the ones who really made this happen. And I'm enormously grateful to them. And I don't understand why I don't have more pictures of them, except that I think we were all working so hard that we didn't have them. So warning, I rummaged through my hard copy pictures and my hard drive, and people will not appear quite the way they do today, which you may enjoy. So this is the story of the genesis of AbSciCon. And for those of you who were there at the time, this will be kind of fun memory. And those of you who weren't, I'm really sorry you weren't, because it was a blast. So this is the account of when the heavens and the earth, and when they were created, and when AMES made AbSciCon. Chapter 1, what the hell is astrobiology? So in the beginning, AMES created astrobiology, and Dave Morrison, who at the time was the director at head for Code S, my boss's boss's boss, said, let there be an astrobiology workshop. The way it actually happened is he dropped by my office, and me being a sort of relatively new hire peon at the time in Building 239, the director at head doesn't just drop by casually, although Michael, I encourage you to drop by anytime. But Dave dropped by and said, do you have a moment to talk? And there is only one answer. And he said, I have this idea for an astrobiology workshop. I'm going on vacation. I want you to find a date. And of course, I went back after he left to my management, and they said, this is the tip of the iceberg. He's not just looking for someone who can read a calendar. And so it was. And so we came up with a date, and Dave saw that the plan was good. And Ro Christian called it AbSciCon. I had this crazy idea we could just call it ABC Conference, Astrobiology Conference. But she kept calling it AbSciCon, and that is what stuck. And so the science organizing committee said, let there be a vault between the presentations to separate the talks from the posters. And it was so, and we called the talks plenaries, and they were in this very room. There was a time that I knew the exact seating of virtually every room at NASA Ames, and I can tell you this room is sort of in the 350 range, and you will see from the press releases later that we were not quite double the capacity. And I do believe at one point on the second or third day, I was sitting somewhere about where Dave was sitting, and Harry McDonald was next to me. I said, Harry, the fire marshal is going to kill me. And he said, relax, Lynn. And here I am today, but barely, because we were so overcrowded, it was absurd. So Harry said, let Ames team with astrobiologists and let headquarters fly above the ground across the vault to the sky, and they did. we had about the right registrants for this room, and word got to headquarters on the first morning that this was the place to be, and people, including Jerry Soffin, called and said, can we still come? And I remember, I think it was Jack Boyd saying to me, please tell me that registration's closed. It's like, I am so far, below this pay grade. I said, I cannot tell headquarters they cannot come. And they came. And that's what was so amazing. They heard that this was the place to be, and they flew out. The advertisement we got in advance was from Spacecraft.com. Keith Cowing was with us the whole way that first year, published the abstracts. He was absolutely wonderful about that. And so you can see here a much bigger list of the Science Organizing Committee and the local organizing committee. We had a lot of diversified funding everywhere from, Mel chipped in and Ames chipped in and headquarters, various orgs chipped in, including Earth Science, Space Science, and so on. And it was just, a fabulous group of people. There's Lynn's name up there, and so on. Now, Dave's idea, I believe, was to have a workshop in astrobiology. I believe at one point when we had lunch in about December of 1999, he said, I'm thinking of limiting this to 50 people. And I believe my response was, we have 200 abstracts. How exactly are we going to do this? And so my conception was this would become the Woodstock of astrobiology, because I was very well aware that there's so much that belongs in the field that headquarters would never fund, partly because of budgetary reasons, but also priorities and so on. And unfortunately, at this point, this includes things like SETI, or the evolution of human skin color. We had Nina Jablonski, who was at the time at the Cal Academy, is now Penn State, speaking about that. All sorts of things that could legitimately be argued were involved in the interaction between the environment and biological evolution and so on. And so with that in mind, let me give you an idea of what it's about. The other thing is we were acutely aware that we did not want it to be the same old, and that we wanted to force people from different disciplines, not just to be at the same meeting and at the same cookie breaks, but literally to be in the same sessions and see how their work seamlessly interacted. And so you can see from the very first one, we actually on Sunday had receptions, and we had some, I think we called them training talks. There was some name for them. But anyway, starting off with the first one, and I still think this was probably one of the best sessions we ever had. We had Philip Ball, who was the editor of the Nature at the time, who had just written a book on life, on water. Having a whole session on water, the sine qua non of life. And so we went from sort of what is water in the universe, with Philip Ball talking about it, to habitability of Europa's crust and ocean, all the way to organisms being able to split water for oxygenic photosynthesis, which was, of course, one of the great innovations in biology. And all this was put into the morning session. And it was good. And here's the second day, and you can see it's the same sort of thing. I'm really proud of the session, for example, on transfer of life, which included everything from panspermia to exotic species invasions, and what we could learn from that. As you can see, we were somewhat clever by inviting Philip Ball as the first speaker, who was an editor of Nature, because that ensured that we had good press coverage. So we did have coverage in Science, and in Nature, storming the Tower of Babel. It was actually very, very nice coverage, but of course none of that counts if you didn't also get coverage in the Astrogram. And there is very much of a certain parallel to today to what it was like almost 20 years ago. And that's where the what the hell astrobiology come from. And if you read this article, you can see what happened was everything, it was one thing after the next as we were preparing for the first one. And then sort of the night before, I believe it was either Ro or Karen said, we have another wrinkle. President Clinton's landing at Ames tonight. And we were having these opening sessions. And what happened was, of course, Harry McDonald was out in the airstrip to greet him when he landed. One of the participants from France apparently got lost somewhere around the airstrip in his rental car, which happens, except that we had the place crawling with Secret Service that night. And apparently they pulled him over and, questioned him. And this all came from Harry, who was sitting there on the runway. And over the intercom, they heard, we've got this Frenchman here who says he's here for an astrobiology science conference. And Harry said, what the hell is astrobiology? And so again, that pretty much summed up that first AbSciCon. And so the fact that the vice president's here today is actually, couldn't be more perfect. So we did all that, and it, it was amazing, and I'm very privileged to have been part of it. And on the very last day, the local organizing committee rested until 2002, because as we all know, no good deed goes unpunished. And so we were already in the works for the second one in 2002. There was a thought of having it every year. And I talked to Barry about this, and I believe Dave, and I think the comment was over my dead body. Dave may have had a clever rejoinder to that remark, but I have forgotten what it was. Maybe that could be organized, but anyway, we did agree, Barry and I, to have these alternate years so that people were not coming, because we also felt that since astrobiology is a multidisciplinary conference, people should also have standing in their own home discipline in order to really be able to contribute. And so you can see Ro was on this always. Hi, Lucille the Great. She's got everything organized months before this is even going to happen, color-coded. I mean, I cannot tell you how fortunate we were to have, again, Ro and Karen and Joe and Shirley, some of them who are up with me, and we still have your room painted mustard yellow in your honor. Anyway, so we were then ready for this AbSciCon 2002, which I believe we just called the second one at that point, again in April, but we had clearly outgrown this room. There was a good chance that the fire marshal was going to have me executed at that point. So we moved to the hangar, which many of you will recall at the time did not look like this skeletal modern architecture structure, but it would actually had a skin on it, but it was so big it had its own weather system. There were pigeons that had lived there literally for generations. You probably could have, Darwin could have done the whole finch study in the hangar. So we meant that we actually had to put up a tent so that we didn't have the weather system. And so this became a very big thing. We ended up having to hire big production companies and so on. And there's Scott helping at the beginning, and we had the younger Jill Tarter, and I don't know how got Scott in twice, and Harry micromanaging it, looking somewhat nervous. Presumably I did what he asked because he's smiling there. And of course, Barry was heavily involved in this, and absolutely fantastic. I think he kind of enjoyed AbSciCon because those were his off years from the NAI meetings, and so we just went wild. Again, it was pictures from the hangar. David Wynne Williams, some of you remember, was heavily involved, and tragically was killed by actually by a car right before AbSciCon, which sort of cast somewhat of a pale. So there was a little memorial for him because he had actually been on the organizing committee. He was at the British Antarctic Survey. There we were in the hangar eating, and there was the poster session. So this is, this ended up becoming a very, very big event very, very quickly. And so we were also able to attract various things. For example, Stephanie Langhoff was able to help us arrange a Klein lecture. And we were very lucky because Chuck had just died a few months before to get Alex Rich the first year, who has also since passed away. And we also had the Burgess Medal awarded to Emery Friedman on behalf of the Burgess Manual Trust. So again, all these places saw this as the place to be. We had sessions on things like science and society. We had a sci-fi night, which Keith Cowing chaired with Paul Davies, who had just written a book on how to build a time machine. And this is actually my favorite picture of the whole presentation. There's one of our aliens fondling Bruce Jakowski. And the big event, though, and because we had Henry G. on the Science Organizing Committee, and he's an editor of Nature, he had tipped me off that we had two papers in press in Nature, one by Bill Schopp, who we'd already invited to be at Planetary for totally different reasons, and Martin Brazier, who said that Bill's interpretation of the earliest fossils was wrong. This took an awful lot of finessing to get everyone happy. There's Henry with the publication in Nature. We were back and forth, and even know, Dave, if you know this, but I was somehow in the middle of emails with the Oxford Public Affairs people, and my favorite comment was, "We will not have our man put in some dark, dingy corner." I may or may not have I mean, I thought, here's a university that's over 1,000 years old, and they're blaming us for putting people in dark, dingy corners. And I may have even said something like, the only one in a dark, dingy corner right now at Ames is my technician, but I may not have. Anyway, we also, once again, got great press from science. And so you can see, you know, we did have these numbers there. It was amazing. We were building and building. So we'd started near 600, over 700, and we were already running for 2004. I'm not going to spend much time on it, but one of the great highlights was that's when we were starting to get results from the Mars exploration rovers, and we were fortunate enough to get Steve Squires to do a special event. So what you see here is my email to Scott Hubbard, who was by then center director, who needed highlights for his Monday morning telecon. And so quickly the bullets were that we had 700 registrants, which means people we actually caught to pay. But in fact, we probably had close to 900 or so because of Steve Squires coming, because all of a sudden we had a huge influx of press, not to mention the Ames people who are really hard to keep out of a room, whether they've registered or not. So we had attendees from over 38 countries, over 400 abstracts, plenary talks, big questions of astrobiology, world-class speakers. We had a session on astrobiology specifically in Spanish that could be taped and used for classrooms where we had, there were students that didn't have a lot of background in English. Steve Squires, of course, he brought in well over, it said 800, but I suspect it was closer to 900. A big debate, discussion on terraforming Mars. We had Arthur C. Clarke in by telecon, another sci-fi night with Andre Bormamis, who was the science director for Star Trek at the time. Code S, Y, and U at the time in the Mars Program Office and the NAI all supported us. Various other things, educator workshops, museum workshops, ethics of exploration. We had people from the press on a panel, including CNN, World News Tonight, and so on and so forth. David Perlman was here. We even had Australian Broadcasting Corporation, Cambridge University Press. It went on and on and on. So you know, it was something that we were all really excited and extremely proud of. But at some point we thought, you know, it actually would be kind of cool to go back and forth across the country. And so we started preparing for 2006. The fact that there's Volvox in the picture as a planet suggests that I had some involvement. And so that 2006 was then in Washington, and that was the last, to be honest, that we saw of it at Ames. And so that's how AbSciCon was launched. That is it. And so that this doesn't just become sort of a retrospective. One of the big questions, of course, has always been origin of life. And one of the great techniques that has really come along in the last 10 years or so to help answer these questions is synthetic biology. And so I'm one of the organizers of the Build-A-Cell, actually NSF RCN, and we will be hosting the next meeting at NASA Ames, January 31st in 2020. So I hope that some of you might want to come to that. So thank you very much for this opportunity. Thanks, Dave. Okay.
Okay, so this is, I think, as we're going along more and more, you're seeing how these wonderful things that have happened, creating a future. And obviously, AbSciCon carries on quite well. And so I think over the next several talks, we're going to see other examples of how, you know, the future has been created in many positive ways by NAI and the astrobiology program. And in the spirit of that, Greg Schmidt now will talk about the NAI model and its legacy. So.
Marco, if you could. Okay. I'll start with a few stories, actually. And okay, yeah, thank you. So I was at, first of all, this has just been a wonderful morning. It's so great seeing all these old friends. It's amazing how long ago this was. It's hard to believe, over 20 years now. And I just wanted to share, first of all, I was at NASA headquarters back in 1996 when the whole ALH 84001 paper came out, and when there was the huge kerfluffle about that and all the excitement and everything. I was working for Arnold Nicogossian, at the time. And so when Dave said his words about Dan Golden's kicking Arnold in the shins, I suspect that happened quite often from the mood that Arnold was in when he oversaw us. One of the things that I'm just so excited about was to have been invited by Lynn and by others to join the astrobiology effort. It was just really a happenstance sort of thing, but I remember well that electric moment when Lynn and I were sitting in Dave's office, Dave Morrison's office. He had gone upstairs for a meeting, and when Lynn looked me right in the eye and said, "I wonder if they've thought of Barry." And we had this very excited conversation. Dave Morrison came right back down, and he went straight back up, talked with, I believe, Scott and Harry McDonald, and they went to Stanford about 3 days later, where Barry was on a long-term assignment. So, okay, all right, great. And one funny story that I wanted to share related to what Lynn said. We, after Barry left the NAI, we brought him back as the distinguished scientist for both NAI and then NLSI. And I remember he was coming to visit, and by this time, of course, we had gotten to be very close friends. And I said to Barry, "So what are you doing this trip?" And Barry said, "I don't know. I'm just going to do whatever Lynn tells me to do." That's what I always do. So anyway, and another thing that I wanted to share as well related to Dan Golden is when Dan Golden was standing in this room, when he came out to officially inaugurate Barry as the director of the NAI. And he said there's going to be 3 things that define NAI's success: missions, missions, and missions. Scott, I'm sure you remember this very well. I think that in looking back at the last 20 years, it has done an absolutely remarkable job at this. When we think of the missions that NAI has influenced and the direction that the agency is going in. When we debated back in those days, it was whether we should include things like searching for water. Now it's accepted that that's part of the scientific emphasis, and it's a matter of how we should search for present and past water and eventually life. And so it's really become a central defining scientific emphasis of all of NASA's missions. And I think Scott said it very well earlier. So fast forward a few years to the time of Constellation, NASA's program under George W. Bush to send humans back to the moon. And the National Academy at the time had been working on a document called the Scientific Context for the exploration of the moon. One of the principals in that is a good friend of mine, Alan Stern. And Alan related this story to me once when I visited him at SSERVI in Boulder. And he said I believe he said he was in the shower one morning having a thought or two, and said, "You know, the NAI has been such a incredible success. Why don't we start a similar institute and focus it on the moon? And we'll call it the NASA Lunar Science Institute." And so that was what gave birth to what is now my institute. Dave Morrison was our founding director, and Yvonne Pendleton was the other director of it. We are now SSERVI. And so what I want to talk about here is not as much about the NAI, but about what it has done for the collaboration and ways of people working together, and in particular founding other institutes. But, okay, all right, very good. Next. So the central word here is collaboration. and collaboration in a fundamentally different way than was done prior to the formation of the virtual institutes. It's collaboration across a number of different boundaries. Dave Morrison shared a great story with that when you went to Santa Barbara and you had all the faculty introducing themselves to each other. And I saw this being in the founding group of astrobiology, particularly at the Astrobiology Roadmap Workshop, which I helped to organize with all sorts of people from different backgrounds that had to not only introduce themselves to each other, but to develop a common lexicon, a common way of speaking. That wasn't an easy problem. Because you have people from different backgrounds, backgrounds such as you see here that are really more of impact to SSERVI, but quite frankly, astrobiology demands an even broader background. If we could advance to the next one. And so what is a virtual institute? It's a distributed organization geographically, and let me pause for just a second and mention that in speaking with Lynn at the break, she said that actually Keith Cowing was the one who came up with the term Virtual Institute. Keith actually shared an office with Lynn when he was out here in the forming days of the Astrobiology Institute back in 1998, and there's a lot of stories there that I won't share. So, so anyway, it's a virtual distributed organization that is fundamentally interdisciplinary in nature, and this model. Scott said, use the words, it's an experiment we expected to evolve. And I really like those words because I remember feeling the same thing at the time. You know, is this going to work? No one had ever done this before. We should really emphasize this. This was the first time that, you know, where we had taken such a wide variety of organizations and expertise, brought them together, but then also had the additional difficulty of geographical boundaries as well. And so what we attempt to do with virtual institutes, NAI being the formational one, but there's now 4 of them, is to eliminate those geographical constraints with the help of the IT that is such a prevalent thing around us here in the middle of Silicon Valley. And then we also take the unique characteristics of the selected institutions that are, as several have said, obtained through a peer review process, and take advantage of those institutions, of what they have to offer. And so yes, as Dave Morrison pointed out, perhaps we could have eventually established a successful astrobiology institute here. I would argue that it would not have been as successful as NAI has been, for the simple reason that you have the best and the brightest from across the country that are able to apply, able to stay where they are, able to have all of their students, all of their facilities, and bring all of that to bear on the problem. We have taken advantage of that at NLSI and SSERVI, and the NAI has just done a marvelous job with that. Let's go to the next slide. So I'm going to skip over this in the interest of time. I only have a few more minutes, so let's go to the next. And so you've heard about the NAI. Next slide, please. The NLSI was the next virtual institute established in 2008, and I remember, as I mentioned, Dave Morrison was the founding director of NLSI. He brought me on, I think that for the main reason, since I had been helpful in establishing the Astrobiology Institute and was familiar with virtual institutes. And I fully expected this to be maybe a 6-month or 1-year job. And here I am 11 or 12 years later. I guess that's not uncommon. And so we, in developing the NLSI, we really relied heavily on the Astrobiology Institute. The model in some cases is slightly different, but some of the things that I can really give credit to NAI for are, for instance, Lunar GradCon, which is something that is very, very similar to what NAI has done with its graduate students. This is something that is developed. It's an annual conference that we support that's developed and run by grad students. All we do is come in and say a few good and hopefully motivating words and support their travel. They do everything else. And we've gotten some just remarkably positive feedback over the years. Another thing that I can say in a very positive way as talk about is the focus groups that Barry Blumberg and others formed at the NAI. And so for us, for instance, we formed an ALSEP focus group. That's the Apollo Lunar Surface Experiment Package, and the head integrator of that package, of those packages for the various Apollo missions, had talked with Jack Schmidt, Apollo 17 astronauts, and said, so much of this data has been lost. How are we going to retrieve it from people's garages and wherever else it might be? And Jack directed him to Clive Neal, who directed him to me, and we established a focus group, and we retrieved data that I am quite confident would have been lost forever. And so we formed other focus groups as well, and I think that we've done a remarkable job resting on the shoulders of a giant, and that giant is the NAI. Next slide, please. The NASA Aeronautics Research Institute was formed here at Ames a few years back. It has new leadership recently, and it's doing a number of very exciting things in helping develop new technologies for air travel. Next, please. And the last of the virtual institutes to form here at Ames is the Small Spacecraft Systems Virtual Institute. It has, like NARI, have a slightly different business model than ours, but the fundamental nature of virtual collaborations and teams across the country is the same. Next slide, please. And so for us, we have transitioned from the NASA Lunar Science Institute to SSERVI, Solar System Exploration Research Virtual Institute. We basically now own all of the objects that are relevant for human exploration. Next slide. And I think that at this point, since I have just about 30 seconds left, I'm just gonna stop. I do want to acknowledge a couple of other people who had really important roles in NAI. One of them is Larry Karoff, who chaired the committee ahead of Scott becoming the first director at NAI that was studying how we ought to do this weird thing called a virtual institute. And under his guidance, Lynn and I and Dave were all on that committee. We came up with a number of fundamental ideas for the structure of this. And so we owe Larry a huge debt of gratitude. And another one is Tom Pearson, actually, at the SETI Institute. And Tom came a little bit, Tom's big role came a little bit later when astrobiology's budget was threatened and got a cut of about 50%. And I don't share this story often, but Dave Morrison and I went to Tom Pearson because Tom wasn't bound by the shackles of the Hatch Act, like so many of us civil servants are. And we said, Tom, here's what's going on with astrobiology. And Tom went and did a heroic effort employing folks like Barry Blumberg, who'd left the civil service at that point, and others, and fought back and was eventually successful at bringing the astrobiology budget back. There's many others that I don't have time to acknowledge. Bottom line, what I want to do is let everyone know that despite the NAI going away, and I personally hope that it's reborn, given its major impact over the years. And I agree very much with Carl that there are many, many things that would not have been done otherwise. But regardless of that, 3 virtual institutes, as of now, perhaps more in the future, have been born and are thriving. And it's really because of the NAI that has happened. And so I owe the NAI a debt of extreme gratitude. So thank you very much.
Okay. We've got 4 talks coming up from PIs who've played a significant role in the NEI, the actual execution of the science and all the wonderful things that you've heard about. And given the thousands of publications and hundreds of people that have been involved, this is going to be a challenge to fit into the next hour, but we'll give it a try. And I guess I have to lead by example here, since I'm talking next on the Charter NAI, AMES NAI team. And I'll just try to give some highlights here to convey the spirit of it. Is this yours? No, that's Scott. That's me.
Yep.
Okay, that's a familiar sound.
Yeah, sorry about that.
Okay, so this is the AMES team under my leadership as PI was for 15 years from the inception of NAI up until about 2014 or so. And I just want to hit on some basic principles that we tried to follow, and then some of the major findings and maybe how that sort of points to the future. In the late 90s, based on everything you've heard, there was this incredible challenge of taking these different disciplines and trying to make it an interdisciplinary science. And so each of these modules that you'll see here, in fact, I'll show on the next slide, we really sort of focus on this concept of early habitable environments as a meeting ground for the astronomers. What do they have to contribute? Planetary scientists and biologists and paleontologists. And basically, as exemplified by this, we have the origins, evolution, and distribution of habitable planetary systems, and something that the astronomers could also contribute to. Then basic molecular biology, origins of life in the cosmic and planetary context, coming from that direction, and then sort of the geologic, paleontologic angle shown in the lower right, all with the intent of trying to understand origins, evolution of early habitable environments, and of course of life. And so early on, in the early years of our participation as a team, we really tried to take each of these supporting disciplines and try to work on aspects that bring them towards this common ground of early habitable environments, origins of life, and that kind of thing. Next slide. So these are the teams. Of course, Lou Alamadola team was just a wonderful addition to this. Cosmic distribution of chemical complexity. There's a lot of organic chemistry out there, but what aspect of it is most relevant to astrobiology? Disks and the origin of planetary systems. You got to take all those ingredients and actually process them through this incredibly complicated thing called a protoplanetary disk, which I came to appreciate over the years, into actually forming planetary systems, and particularly ones that include a habitable planet or two. Mineralogic traces of early habitable environments was once that you have this record on a planet like our own, how could you interrogate the materials of the crust of that planet based maybe on mineralogic traces and other things to infer the early environment and also the presence or presence of life? Then origins of functional proteins in the early evolution of metabolism, that's Pohorile's group, very much interested in how you just go from the simple monomers, and there's been decades of research before that you can come up with interesting monomers, but how do you get them together into bigger molecules, and most importantly, how do you get the molecules to play nice with each other to form the earliest metabolism? Education and public outreach, of course, has been emphasized with something that we incorporated, and you heard many of the types of programs that we participated in for that. And I'll give one example later on. So next slide. Okay, so these are just some quick highlights from a lot of work over a lot of years. The major thing I think coming out of the Alamandola's group, the astrochemistry group, is this understanding the abiotic formation of molecules that are embarrassingly close to what we would call biosignature molecules. And in fact, amino acids, obviously, amphiphiles, which make membranes, quinones, which are important in redox reactions, and then nucleobases, like they actually found in their synthetic experiments under cosmic conditions, as illustrated by the apparatus in the upper right, actually made uracil, and also showed that it was very hard to make pyrimidines, which makes sense because most people think pyrimidines probably came a little later in evolution. So, that's just one highlight, and you're going to get the benefit of hearing Scott Sanford talk more about this in the extension of our team, which was supported after 2014. So I'll just move on. If we can go to the next slide. The next group was originally what started in the original team, had a number of different aspects to it. Rich Young was the original sort of leader of that group, followed by Sandy Davis. And this was the sort of follow the water in a literal sense from just we have the protoplanetary disk. How does water partition given the dynamics of that disk? As the disk and dust, as the disk is removed and you form planets, how do you navigate the water through that, giving rise to the planetary inventory that you end up with. And then, of course, a lot of modeling, Jack Lissauer and others, given the powers of computation here at Ames, and I should have mentioned that also in all of this, is that a key aspect of astrobiology was always identified as being important to have at Ames because of the computational capability. And this group here in particular really took a lot of advantage of that. A lot of simulations by Jack and others basically showed that the process of planetary formation is so stochastic that you really can't predict where you'd have habitable planets. You just have to go look. And of course, that became an intellectual justification for the Kepler mission. There's just no substitute for actually going out there and seeing what the distribution is. A lot of that based on that. And of course, a lot of the results coming out of the database from exoplanets, Greg Laughlin at UC Santa Cruz, who was on the team, came up with this idea of having a database where you could process all these data. Once you got the planet and it's halfway habitable, Kevin Zonley then talking about what was that early environment given everything that was going on, an environment that could sustain life. So a lot of aspects in this group. The next slide, if we still have a slide changer back there, there we go. In the earlier few years of our team, maybe the first 5 to 10 years, we had a fair effort on microbial mat biogeochemistry. Now, microbial mats may sound esoteric. You brush them off your teeth twice a day, and you find them growing in places where the water is a little too moist around the perimeter of the house and all that. But actually, they are the oldest, most widespread ecosystems known in the Earth's rock record, these biofilms, and particularly the photosynthetic ones. And they probably hosted several key evolutionary events. They sort of, in many ways, were sort of the microbial Garden of Eden. They've had a number of things that allowed them to persist quite well under adverse conditions. The key thing is that the environment inside these mats is quite different from what you would infer from the outside or you could maintain in laboratory cultures. And that's the environment in which their physiologies developed and the selection process acted. And so it was very important to drill into that. And we're able to do that with a team that would not have happened in the absence of the Institute. The number of people involved and all the logistics and everything. And of course, the punchline is that they create biosignatures that could be preserved in the rock records. And in the lower left, you now see on the lower, the gray image there is actually a ratted surface of a rock at Meridiani Planum at the same scale as you're seeing a microbial mat. We are now at the scale of observation where we could see these darn things on Mars if they were there. We haven't found them yet. But, these are really relevant types of features to engage in our search for life on Mars and elsewhere. Next slide. Tori Holler came up with this idea about just sort of the energy balance as a way of assessing habitability. In other words, it's not just a black and white thing. You got liquid water, you don't, and that's the end. It's really, does the organism have enough energy to cope with all the environmental challenges in order to be able to persist and propagate? And so he not only came up with sort of a numerical way of approaching this, working with others, looking at how life could exist at extremes just based on this energy balance for life, but also how the evidence for that might be expressed in the rock record in the form of minerals, organics, and other types of things. So a nice systematic model that's now really being taken up by a lot of people going forward. The next slide moves on to Andrew Pohorili's stuff, and this group achieved so many things I couldn't possibly hope to represent them in one figure, but I think some of the key things is that they found that the functional proteins can be actually quite small, and you're approaching the size of a peptide that you might see being able to jump to from amino acids that we've already seen in amino acids and in meteorites and elsewhere. And so this gap between what we know can happen abiotically and what becomes functional is actually not as big a gap as we once thought. And this applies for enzymes, also applies for membrane proteins, did a lot of elegant modeling on how you get functionality across membrane proteins and so forth and so on. Just a lot of aspects here that I really don't have a lot of time to summarize in detail, but the point is that this gap between what we know can happen abiotically in ancient environments and maybe the minimal structures required for functionality, this gap is being narrowed by, again, very computationally intensive work by Andrew's group. Next slide. And then just a wonderful example of how, and again, using ab initio modeling in the computation center here, showing how a peptide can insert itself into a membrane, become a transporter, and achieve an active state. And that these don't need to be very big. In fact, the reason that the proteins are as big as they are today is all about regulation and sophistications that developed later in the history of life. Next slide. And so I'm just going to also make a pitch here about education outreach. We're engaged, of course, in a lot of the programs that you've already heard described. But one thing that we did that I think is a little unique is at the high school level, this thing working with Lassen Volcanic National Park and Red Bluff High School, a student intern program where, yeah, we teach them about astrobiology, but what we really teach them is critical thinking and the scientific method. And it's amazing when you just sort of get them into it with a field trip, making observations, doing follow-up stuff in the lab. Kids are scientists, and they're born being a scientist. They're just absorb information. The key is to keep that excitement going through junior high and their high school years. And I think just getting them into the scientific method, because if nothing else, if they're not astrobiologists after they leave high school, you want them to be critical thinkers and citizens who actually use facts and the basis for their decisions in life. So I think we've had a lot of good legacy stories coming from students. This program's been going on now for 10, 11 years. Next slide. And so, of course, a lot of contributions to flight missions. I really don't have the time to go through this, but each one of these aspects is sort of highlighted in the orange text, has had a lot of participation. There's probably more under Mars here because that's stuff obviously I know the most about, but I'm sure the astronomy group could add a lot of embellishment to what I see down here. But we really have had a major engagement with a lot of different exploration venues. Next slide. And I just want to say, in terms going forward, we're envisioning here at AMES, with the computational capability and also us being a NASA center, that service to the communities is what we do, sort of parallel to what a university professor does when they teach. We provide community service. And I think this whole idea of databases and the need for that going forward is just becoming more acute every year. A great pioneering example of this is the POD database, and maybe Scott will talk more about this, but this is now a publicly available thing, which is really important for just cataloging all the detections of organic matter out there, all the modeling that's been in the labs, to validate the spectral features. And this is available to the public. There's literally been, this slide is from 2010, and I think already there's been thousands of hits on that website. And so it's just a real legacy thing. And I also then, if you go to the next slide, want to point to the Origins of Life focus group that Andrew Pohorili maintained with John Peters for a while there. And he came up with one thing, which was sort of an online debating tool or discussion tool called the Hypothesis Browser about origin of life. You can imagine there's a lot of hypotheses. And this is now being drawn into this AHEAD database, which is an astrobiology database to try to systematize and make accessible work that's been done, funded by NASA, but maybe other work in the name of astrobiology. and having this idea of an online discussion tool is a very important part of that database. So I think a lot of the science that's been done has legacy going forward, certainly in terms of impact on missions, but also just how we organize the community and preserve data and interact in the future has also a very important legacy. So with that, I will finish. How are we doing here? I think we're good. Anyway, thanks. So, and speaking of NAI or AIMS teams, the reincarnation of our team as of about 5 years ago is going to be presented by Scott Sanford, NAI at 20, prebiotic chemical complexity.
Okay, I'll dive right in because time is tight. So I just should mention that Dave already mentioned that we were a part of the first Can 1 team from AIMS, and that's in large part because Lou Alamandol and I had set up the astrochemistry laboratory. So at that time, he was called an astrochemist and I was called an astrophysicist, and we were largely, we did fundamental research, but we were also largely working on supporting NASA projects and programs, and in particular, at this time, the field of infrared astronomy was really growing by leaps and bounds, and we had, over the course of 15 years, evolved from a community that often talked about how there would be few or no molecules in space to actually finding them all over the place. And so two areas that we were particularly focused on was trying to understand emission features which were being seen all over the universe, which we subsequently were able to demonstrate are caused by polycyclic aromatic hydrocarbons, and Dave just mentioned the POD database, which is one of the products that came out of that. And in addition, we were very interested in looking at star formation regions and dense molecular clouds where we were seeing ices, and people want to know what were producing all these features. And so we were running back and forth between telescopes and the laboratory trying to match these spectral features and figure out what was going on. And we did identify a number of ices that were around protostars that were forming in dense molecular clouds. And one of the questions we asked ourselves is, well, we know radiation is in these environments. What does that do to the ices? So we actually did experiments to try to understand what happened to ices when you irradiated them. And what we found is you can take an ice that's consists of just a few simple molecules and irradiate it and produce literally thousands or tens of thousands of new molecules whose complexity is up by several orders of magnitude from the starting materials. And so that was fascinating, and we try to understand these residues of goo from the standpoint of astrochemistry and astrophysics. We wanted, so we figured, let's try to figure out what the most abundant things are. What does this residue mostly look like? But that's a really hard task. You've got a million lines in there, narrowing down to one and figuring out what it is a very difficult way to proceed. So by brute force, we did in fact manage to identify at least some of the things that were in the residues, including things like amino acids, which was fascinating. But the way we approach things changed when the field of astrobiology came along. And one day I remember someone came in and informed Lou and I that we were now astrobiologists. I used to joke with him that, we went to bed one night as an astrophysicist and astrochemist, and we woke up to be astrobiologists. And I wasn't really sure what to think of that at the time. I think I had a slightly negative attitude about that. I've never taken a formal biology class in my life. Probably my first response was, what the hell is astrobiology, which we've already heard. But on the other hand, on reflection, we realized that this really had many advantages. By the way, so if it's clear that I was surprised to be an astrobiologist, you're not surprised that unlike many of the other people who have spoken here, I have very little to do, essentially nothing, with the invention of astrobiology and setting up the institute, except I did do one key thing unintentionally, at least a decade before all of this, and that is in the astral summer of 1984-85, I was living in a tent as part of the ANSMET field team collecting meteorites in Antarctica, and we picked up Allen Hill's 84001. And I've helped collect probably several thousand meteorites over my life, and I don't remember picking most of them up there. But this one I remember. We knew right away this rock was weird. And I even remember that we discussed the possibility that it was from Mars, although the general consensus was probably not. And when it went back to JSC, it was misidentified as another class of meteorite, and everyone sort of forgot about it for about 10 years. And then, so when we found this, we knew it was weird. We had no idea what a fuss it would cause later on. So that's my one claim to fame, possibly, to getting the field of astrobiology rolling. So one of the immediate advantages of becoming an astrobiologist was that we suddenly were able to join with Dave Desmarais, who was setting up the AMES team for Can 1, and becoming part of the NAI was just a real boon to us. And I've listed several reasons. First of all, of course, we got to work with Dave. Anyone who's done that knows that that's no small thing. And then, of course, it provided us with resources that were on timescales that were made us much more stable. But two things I really want to highlight is, one is it got us to interact with people we never would have talked to otherwise. And this resulted in a lot of changes in what we did and how we did them, and really modified our approach. And in particular, for item 4 there, it's a difficult thing to look at that spectrum of all those peaks and try to figure out what any given peak is. It's a much easier project to say, like, okay, maybe all these peaks aren't equally interesting. Let's see if we can find which peaks in there are interesting. So, for example, do we make quinones? And you can go look for those, and they may not be the biggest bands, but they're astrobiologically perhaps some of the most interesting bands. And so that really gave us a way to focus our work that allowed us to make progress much, much faster. So at the bottom here, you see a picture of what happens when you take our residues and you dump them in water. Our residues contain some amphiphilic molecules which spontaneously form into vesicles when you put them in water. They're not the kinds of lipids that are in modern cells, but they may well have been the kinds of molecules that formed early structures on the forming planet that played a role in getting things moving. So we worked with the RC team with Dave for Can 1, 3, and 5, and we're very happy about that. But when we got the original write-up for Can 7 and got together to discuss writing the proposal, it became clear that we were going to have to split up the way it was written. It was I remember being a very sad day. So we took the astrochemistry portion of the earlier teams, continued to focus on the astro portion of astrobiology, and augmented the tasks we were doing to include some quantum chemistry work, disk modeling, exoplanetary research. Some of this was an extension carried forward for what had already been in the earlier Can teams for AMES. And ultimately, we were selected. And I have to say, when I was selected, that was, for me, a very bittersweet outcome. I was happy to have won, but I wasn't happy to have left Dave's team. So here are the members of the team. I'm not going to dwell on this in great detail. The center of mass is, in fact, here at AMES, but we do have members who are at other areas, and even though they're all listed as being part of a particular work group, as I'll show you in the next slide, I believe, we interact at a very deep level. So I was sort of interested to see Dave's slide. He had a slide that has actually almost the same structure, something at the top and two things at the bottom and a thing in the middle. He used boxes and circles. I used hexagons because that's the shape of paws, okay? So that's a better shape. So, so the top one are some tasks that involve modeling of the formation of planetary systems through protostellar disks, and also working on data from exoplanets. The lower left is lab astrochemistry, and the lower right is quantum chemistry, and all of these have their own tasks, but they interact with each other in very specific ways. So, for example, the modeling of disks informs both of the other two boxes what kinds of environments you need to be simulating, what temperatures, what pressures, what compositions, what stuff in the gas phase, solid, whatever. Similarly, the two tasks at the bottom are coming up with reaction paths, reaction efficiencies, infrared spectra, and these can all feed back up into the model. And then the quantum chemistry in the lab interact by us identifying things. I'm trying to figure out what the reaction path might be, suggesting additional experiments to do to try to sort out the possibilities, and going back and forth until we sort that out. So Dave showed that we can make uracil, the nucleobase uracil. We can actually make all of the nucleobases and lots of variants of those. And an interaction between those two bottom teams really played a huge role in sorting that all out. And of course, all of this can feed into NASA programs. The the disk models to inform infrared telescopes and radio telescopes, what kind of molecules to look for, what kinds of predicted distributions of molecules you have. They can help interpret data that's being taken. If you're going, instead of outward, come more back inward, I work on sample return missions like Stardust, OSIRIS-REx, Hayabusa, Hayabusa 2. And this work really tells us something about the kinds of compounds we need to be looking for when we get return samples back from primitive bodies in our solar system. And these are bodies that have preserved the original material from which the solar system was made and tell us an awful lot about how things got started. So I'm just going to talk about a couple of these tasks. The disk modeling has gone beyond the normal models where you just try to figure out how you get all the dust and gas to clump together and make planetesimals and then get those together to make planets. Our team has been adding reaction schemes to this so that while everything is moving around, clumping, dissolving, vaporizing, condensing, we're also following the creation and destruction of molecules and can track where they go. So this means that you can, at the end of it, when you think you've got a planetary system, look back through the time and ask yourself, if I'm getting samples from objects in the solar system, what do I expect at this distance from the sun? Is it matching the models or is it not? This is one of the areas where I have the most regret that we're not going to be able to compete for another round, because this modeling effort is really coming to a head here, and I see a lot of potential for it. So I'm personally committed to trying to figure out how we keep this task going in the absence of the NAI. So Greg Laughlin has worked a lot on exoplanets. He's looked at the statistics of exoplanets and found he's just always amazes me coming back with interesting and amazing things. So he has used databases to demonstrate that our solar system is a bit odd, that solar systems that have a Jupiter at the position of Jupiter are not common, but that systems that have Neptunes near the Neptune's orbit are common, and one of the consequences of those is that they ought to chuck a lot of planetesimals out of the forming planetary system, so that interstellar space should be full of a lot of cometary bodies that are wandering away from their original point of origin and could, for example, be passing through our solar system. And we now have detected two interplanetary comets that, or sorry, interstellar comets that have come through our solar system. Also, he's done modeling that puts constraints on where, if Planet 9 is out there, where it is. So Dave mentioned a few of these things here. I won't talk about the formation of the nucleobases because that was already mentioned, but in the past couple of years, we've demonstrated that in addition to amino acids and quinones and amphiphiles and nucleobases, we can also make sugars and sugar-like compounds, sugar acids, sugar alcohols. And we've been exploring the consequences of re-irradiating our residues, because this should have happened in the protosolar nebula, to see if we produce materials like many of the materials found in meteorites. Most of the carbon in meteorites is in not soluble form and interesting things like amino acids, but it is something called insoluble organic matter. And that has some very interesting properties for isotopes, gas trapping, and so on. And we're seeing if our residues can reproduce any of that when they're re-irradiated, and all of this stuff will be put back into the models. We're also looking at how gases interact with solids, not just irradiating ices. And so here are several examples where Andy Madioda and his group have exposed minerals and organics to gases or radiation and seen what happens. And so, for example, he's been able to demonstrate that you can deuterate things from the gas phase and gas grain interactions, which has been a model that's been around for a long time. And also that you can add hydrogen to PAHs and turn them into aliphatic components. So this little equation in the middle looks simple, but it's really a nightmare. But we have people who've mastered it. And so they can do lots of things with this. One they can do, for example, in the middle slide there, is to explore how one goes about making aromatic hydrocarbons, which we see everywhere, starting from acetylene, which everyone's always thought was one of the main ways it might happen. But how you get there is obviously complicated, and the figure shows that. Down in the lower right, they can calculate a very high resolution rotation spectrum from gas phase molecules. And by working back and forth between lab data and that, they can get to the point where they can in fact predict lines for many, many different vibrations at for the molecule at many, many different temperatures. So they produce line lists that contain literally millions of lines. So this is a big database thing right here. And these are things that are of particular interest for example, to people who want to study exoplanets. And one of the ways you're going to tell whether there's life there is to look at atmospheres. And so if you can do that, you need to know what the rotational gas phase lines of lots of different molecules look like. And so they're very active in that area. So sort of to conclude, the combination of all these telescopic observations, modeling, the laboratory simulations, and everything suggests that abiotic organic synthesis is common in space. I mean, the environments we're simulating are sort of universal, so whatever happened here probably happens all over the place. And so if you form new planets, you can probably count on this stuff coming. Now, most of the components I mentioned we make are complicated compared to, let's say, water and methanol and some of the things we start with, but they're not complicated compared to the kinds of biology, biochemistry in life. So the analogy I often use is if you think of life as being a very complicated Lego castle, then we've demonstrated that the universe really can easily make Legos, okay, and that these Legos are going to be distributed all over the place. So of course, one of the next steps then is how do these Legos start to assemble, and that's another very important part of astrobiology. But because these processes are universal, I think you can really count on this stuff being there. So in that respect, the idea that life might be common in space doesn't seem so crazy to me. And I'll end there.
Okay, moving right along here. We're now going to hear from Natalie Cabral, the NAI at the SETI Institute, Astrobiology Exploration on Earth and Beyond.
Thank you for the opportunity of talking today. When you asked me to talk about the NAI, the SETI Institute, you also mentioned that you would like to see a transition towards, what the future might look like. I heard a number of things this morning. I fully agree with the mission. I think that really our community is the best to actually advise what a future mission should look like. And so I wanted to give an example of what I think is applied astrobiology to mission, and talk about some of the aspects that we have been working on with my team, which is basically to look at upcoming missions to Mars and trying to understand if, because we are now going from the characterization of habitability that we have been doing for the past 2 decades to the search for life, there are incredible changes in scale. And in what we wanted to understand is that, are we prepared? Are there critical knowledge and operational gaps in the missions that we have right now? And if there are gaps, can we help develop methods and tools to fill them? And in the process, we're also advancing the operational understanding of the technology that's already there, but also maybe informing on new instruments and how to use them to fill those gaps, and also inform the collection of sample, how to catch the best sample. So the baseline is that for us, acting as a body of the NAI community that would make not only recommendation, but also help build the future. I'll get to maybe what I would suggest for the future of the astrobiology community, only if you have time now, maybe in the afternoon during the discussion. So we needed to start somewhere, and places where we can address this knowledge and operational gaps is really to understand, what drives the distribution of life in a polyextreme environment. And Mars was a polyextreme environment right away early on, like the Earth was. And do we have currently the necessary data from the orbiters and rovers to support what I call a reason versus random search for biosignature? You can go with a rover and make traverses for a long time and just pick some rocks that are interesting or look interesting, but you can also go about it statistically. So what we want is to improve the odds of catching critical sample. So summary number from our project, we went through about 10 field sites in pretty much everywhere you can think that you have analogs of Mars on Earth. We have collected tens of thousands of spectra, thousands of natural samples. I think this is a very low number here. We will be probably much higher than that at the end of the mission. And also dozens of synthetic samples were produced. We went through some complete integrated analysis from orbit to the lab, to the ground to the lab. For 14 of them so far, there might be more. So we are following through the whole scheme of what we are hoping to be an astrobiology mission leading to a Mars sample return mission. And we're also contributing to the libraries of physical samples and images. One of them is being at AMES here, which is the Raman library, which is led by Ginny Gulick within our team. So I really put that team together more as a task force trying to solve a question, trying to understand mission issues. And these are the expertise of the people in my team. And to the right, you see the name of those. I'm not going through them, but there is a deep connection between AMES and the SETI Institute. And all of those were not showing that they are from the either the SETI Institute or NASA AMES or former NPP or NRCs at AMES, pretty much all of them. So what we did as well was to train the next generation, and we're still doing it, of students and young researchers. These are the number. They have participated and still participating actively to the project, from the design to, as you were mentioning, Dave, having new ideas, just coming from a fresh start. And because they are helping in different ways of the project, connecting the dots where sometimes we don't have time to do so. So the baseline is that what we want to do is to connect the megascale, the orbiter scale, to the ground and to the microscale. And right now what we have is a lot of information about habitability, which is driven by the environment, but we have much less information about what is a habitat, which is driven for life and coevolution. So first of our goal was really to understand, where do we start to go from, well, here's the threshold of attention. You got my attention. This is a good place, down to the potential threshold of detection. Here is a place that look like potential habitats could be there, and then getting to a place where is really where your habitat is being confirmed. So we have been doing a lot of work with orbital imagery, with drones, etc., to address the different thresholds. We have identified them, and when we were there, we understand that right now we don't have many more orbiter missions to come. So there are some tools and data that we won't have, but what we can do is to improve those we already have and make them better. And so developing those tools meant for some of our teams to develop, to do improvement of imagery using hyper resolution, also developing some hypothesis mapping and probability mapping, and have dynamic mapping. As you are making measurement, your map is going to improve, and you are going to have a better idea of what is a potential habitable environment. And this involves deep learning for image analysis and automated terrain mapping. Some of them can be applied to orbital imagery, like on this slide. Others, of course, can be applied from the ground. And what you see to the left here is some of the results of these image processing. So what you have here, you wouldn't have right now. We don't have this kind of resolution from a drone imagery, but hey, we have drones, right? We are going to learn how to work with them. And not only this is giving you a bird's eye view of your terrain even before you get the boots on the ground, but as you are working somewhere, you can send your drone, get the imagery. Here you have an imagery which is at millimeter scale. So now you are starting to think really habitat. But for me, what connected the dots really on the ground, especially this year, we had the possibility of taking the hyperspectral imager that was granted to Jeff Mersch through a planetary imager equipment at NASA. And as he was taking those hyperspectral cubes, all of a sudden we were able to match some of the spectra with the statistical work we had been doing on some of the feature on the ground. So now you are getting spectra that are connecting the dots between the remote sensing, the microbial habitat level, and not quite yet to the orbit, because we don't have that much resolution yet on orbit. But that tells me that we absolutely need capabilities to do hyperspectral to multispectral imagery at the scale that can match the ground with that. And at that point, we will be able to tell that microbial habitat may potentially be in some places. But we know that because we have been doing an incredible amount of work, statistical work, which right now the missions that we have don't allow. We have big rover making big traverses. We don't have the capability of staying in place, which microbial ecology requires. You stay in place for a long time and you just learn what your microhabitat is, what our scales are. We have done 100 of samples and we have followed through in the field. What we understand is that, of course, colonization is patchy at microscale in polar extreme environment, but there is order in the chaos. And this is, for us, a big news is that those distribution patterns that we found in the habitats we have visited, they are repeatable at various scales throughout the landscape. We need to learn them. When we learn them, when we know that they are, the landscapes start to lit up like a Christmas tree. So what is interesting is that you can do predictable mapping. The thing that is a little bit more constraining is that microbial habitat in polyextreme environment are dominated by local to microscale factor. They are dominated by mineralogy, by texture, by the weather, the local weather. They are not dominated by the kind of data that you can get from an orbiter. And so we will need to learn those things. This is really where we have big gaps. So we also are building predictable maps where to find those biosignature using this kind of methodology. Now I'm going to go on how we did apply the instruments that are currently in missions on Mars with MSL or will be on Mars 2020 and ExoMars, because we had people involved with those missions as co-investigators as well. So we tried those instruments in different settings, and we also produced library as, that can be used by the team in future missions. And so we did that with the Raman leaves, where we can put the organics within their chemical context. So we did that in Chile. We produced a library of other 2000 spectra. It's there. We have some samples related to that. We did the same thing with the ExoMars RLS, trying to start building a library for lipid and lipid standards. We also put on the ground a prototype of a UV fluorescence imagers and showed that actually not only you can detect these organics, etc., but you can tell how much, you know, their intensity and their abundance in the field just by looking at the color of your signal. So this is a work that is being done by Heaven Eshelman. And we also have a number of instruments that are new operational concept. One of them is a direct heritage from Kemen. Here we have Philippe Sarrazin and David Blake who contributed to that. Philippe is working on MapX. MapX is going to be ideal really to give you a chemical map of region of interest in samples or in targets. And another one that I found really very interesting is PIXI. PIXI will and might help see through the caching tubes if there is a core or no core inside, so that you don't waste any time bringing back something that, you know, was not successful. So these are operational scenarios that we can work into future missions. Also, in terms of services, we've been helping deploy some of the coring and drilling systems that will be used by Mars 2020 and ExoMars. And as a result of all the testing that we have been doing, Honeybee, through Chris Zachney, has been producing coring and drilling kits, and those kits are now available to the scientific community, the astrobiology community. Some of them have been used already in multiple projects, and not only NAI project, but also PSAR project, and beyond that to human exploration with the NEMO astronaut training. Some of the coring devices have been used underwater. So for the drilling system, same thing. For ExoMars, we have been using this to help our scientific investigation, but also help the engineer fine-tune the characteristics and the specification of those instruments. And because we had we didn't have way too much time, we have way too much drones, we started thinking about operational scenario, new ones that could be very helpful. As an astrobiologist, it really pains me when somebody tells me, well, we cannot go there because the rover cannot go there, I say, I want to go there. You find a way to get me there. And so sometimes it's difficult for a rover to get to those places that microbes like in polyextreme environment, which are small slope, high exposure, etc. I was very excited when I saw the drone in the Mars 2020 mission. I understand it's going to be a technology demonstration. This time we need to do a lot more. We demonstrated in Chile, in Salar de Paronales here, that we can acquire a sample, bring a sample back to the rover or to a lander, and that was over a 1 kilometer distance. So this is starting to work. There's still a lot more work to do, but here are the fixes that we can, or actually not fixes, but some future research avenues for that. I'd like to just mention some of the science and technology payoff from our team's project. What we've been doing is really develop an optimized strategy to maximize the science return from the upcoming missions, but also astrobiology survey in general. What we do for Mars, take it as a principle. I don't remember whose talk was talking about principle of astrobiology. That might have been you, Scott, or David, but it's really about principle. How can we get, the engineers to understand that there are principles about doing some typical research in astrobiology? You don't look for microbe the same way you characterize habitability. So what we hope is that we have informed how to survey core and cache operation for Mars 2020. Obviously, there are capabilities that are not there yet. But what we can do also is support the teams that will be on Mars 2020 and ExoMars by populating spectral libraries and telling them the type of sample that we found, the type of texture that are favorable to microbes, etc. But also we contributed equipment and facility to the Earth Planetary and astrobiology communities, and that extends way beyond the realm of the NAI. Also because we get together with people working on other mission, the NAI was instrumental for us through DDFs. Through DDFs with teams, the IC World team, and ICIC is here, teamed with ours, and we got a project going that ended up being a winning P-STAR proposal that will develop exploration strategies for Europa and ocean worlds. Also, I think that we have then identified that, hyperspectral imaging as a very, very promising way to connect the dots between orbit to ground or aerial to ground. And that will be instrumental to get us a better idea of where habitats, microbial habitats can be. Also very important is that there is order in the chaos of microbial ecology, and that if we can figure that out on Mars and elsewhere, then we might have a much better chance of finding the good sample. And to me, drones are going to be critical in filling that gap in the coming years. So what's missing now? I think that we are missing data, spatial scale, and spectral resolution relevant to microbial habitat in the current mission and missions that are being planned. It requires a lot more than long traverses. Right now, we are leaving it a little bit more to chance than I would like to see. But the good news is that we can improve what we have. So there are two ways of going about that. You mentioned machine learning and AI. This is a wonderful way. We're seeing on tons of data that we can improve, that we can also automate. And we also need to think about a new class of mission between now, Mars 2020, and Mars sample return, because if there is nothing in between, then we are in trouble. So a summary of what the role of our community could be, I think that we are a task force. We can be applied to be the bridge between the engineering team that are building technologies and telling us, here is a big thing that I built, try to do the best you can with it. I think I'd like to see, us coming up with what is really important, what's needed, and please develop the technology around that and get me to that side that I need to go to. At the programmatic level, I think there is a lot of room for discussion for the astrobiology community in a way that the whole structure of the program and the discussion. Top down might be a little bit better, bottom up in some cases. I'll discuss that this afternoon. And I will mention in passing that I still think that Mars 2020 is mostly a habitability and environmental context mission rather than a life detection mission, in the sense that, again, you know, life detection will require more statistics to have a chance to be, or a better chance to be successful. And that we need more missions. So that is what we stand right now with the project.
Okay, last but not least, Mike Mama, 16 years in the NAI synergy amongst NAI teams and paradigm shifting science at Goddard. My goodness.
Right, so, okay, I'll advance, thanks. All right, thank you very much. I guess I'd like to begin by saying that my involvement with NAI really began long before CAN3, which was the first time we were selected. I served as part of the work, the road mapping exercise in 1998, and then I came to the conference in astrobiology here in 2000, which was mentioned earlier today. And then Goddard decided to propose for both CAN1 and CAN2. However, that was dictated from the top, the center director level. Actually, Jerry Soffin was the mover behind it. And unfortunately, those two proposals were very poorly organized, and they didn't really exploit the strengths of Goddard in the area that was, I think, best suited for astrobiology. And so both proposals went down in flames. And I fortunately served on the review panel for CAN2, of course, refraining from participating in any review of the Goddard proposal. But when I returned, I organized a presentation to Al Diaz, our then director, and laid out for him a plan which would lead to Goddard's selection in CAN3, in my opinion. Part of that plan was that we would focus on the issue of the origin and evolution of organics in water in planetary systems. We already had a lot of internal expertise in the area of cometary science, and I'll talk about that in a moment. But we were weak in the area of asteroidal science, in particular carbonaceous meteorites, both of which comets and such meteorites could deliver organics and water to the early Earth, the barren Earth. And so as part of that, I proposed that we needed to hire 2 new people before we submitted the proposal, so as to show the institutional support for our involvement in the NAI, which is the key element of the NAI structure. Unfortunately, Al bought that, and so we actually chose the very best available person to set up the asteroidal research facility, and that person was Jason Dworkin, who was then a member of the SETI Institute here at AMES Research Center, and was essentially a soft money postdoc. But when he came to Goddard, we actually set him up in a new astrobiology analytical lab, which he became director of. And we funded that lab with internal funds. And so when we proposed for CAN3, we were able to propose from a position of strength in both the area of analysis of these primitive organic materials in both comets and asteroids. And that is really a very important thing, which I'll show a little more detail on in a moment. Now, most of these points here you've seen discussed earlier this morning, and so I won't dwell on those further. But I do want to show you the membership of the first team that won on CAN3. And with these red arrows that you see are basically several people who were heritage from another NAI team. Steve Charnley from the SETI Institute was also, he's a co-I on our team from the beginning, and later became another civil servant whom we hired here. And I met, you can see the red arrow, all the particular names that are starred below the label GSFC are individual civil servants. And of course, Jason was, by the time we actually proposed, one of those. We also had, you'll notice that this is organized in the context of the institutions, and our team is virtual, like most of the other teams in the Institute. And you can see we had approximately 8 or 9 different institutions involved to propose and carry out the science. We also had many collaborators. And again, here I've marked with red arrows the folks who actually agreed to serve as formal collaborators on the proposal. The formal difference being that COIs, of course, get funds. Collaborators might if they really needed them to carry out the joint research. And so here you see on the left the NASA AMES group, which you just heard from Scott, leader of that team, and all four of those folks had agreed to participate in our proposal. On the right, we also had representation from Penn State University team. Stein Sigurdsson was the person, point of contact, but his student, Avi Mandel, a graduate student, came to Goddard and actually completed the second half of his PhD dissertation under my leadership, doing work on infrared spectroscopy of molecules in circumstellar disks and also in exoplanet stars. And then, of course, we had another collaboration with the Carnegie Institution, which continues even to this day. So here you see right away this the miniature of the institute, where you have four NAI teams basically interacting as in a virtual way, but to enrich the capability to do this class of science. In CAN 5, now we're showing both COIs and collaborators on the same chart. The COIs are in red. Again, we shown looking at the red arrows, these are all folks who basically were associates who came from other NAI teams. And now you'll notice that most of them are in red. They've been hired as civil servants by CAN 5. So Steve Charnley is still at Goddard. Avi Mandel is now director of the SEEK Exoplanet Consortium at Goddard, which is again a virtual entity essentially. And he has he's also a PI on the Nexus team under the RCMs. So he's come from graduate student under this NAI leadership up to now a PI himself. Jen Eigenbrod and various others. Stephanie Milam, you all know, I think she was here. She's now deputy project scientist for planets under James Webb Space Telescope and has her own PI class investigations at Goddard in the lab, both the laboratory and also in observations with ALMA and other radio telescopes. So in CAN 7 now, as you already heard, there was a paradigm shift. And the sense was that CAN 7, we got directions in the CAN that we had to actually caused us to fission a bit, and I had to split off the group that was involved in lunar samples because that was targeted by another program. But indeed, you see with now the even more red arrows, we have these collaborations with other NAI teams, Carnegie, and a very strong continuing collaboration with here at NASA Ames with the Sanford group, and so forth. We also had collaboration by this time with Karen Meach, who was a PI from UH and that team, et cetera. So again, you see this tremendous impact of the inter-team activities, which went on and has been very fruitful. Now let's, for our team, the principal question we're trying to address sounds simple. Why is Earth wet and alive? But as we all know in this room, it's not at all simple. Earth formed as a dry body in a dry region of the protoplanetary disk, and after the moon forming catastrophe, whatever water probably had evolved by that time was stripped away. So it was a hot, barren source, but within 250 million years, it was a wet ocean as indicated by zircons, which are associated with formation in wet to high pressure regions. So the issue is, how did Earth get its water? And we think that most of it was delivered by comets and asteroids based on deuteration ratios in various bodies. And then, of course, the secondary question is what prebiotic organics were delivered and in what quantities and so on. So in order to address these questions, we have a whole variety of tools and so available to us, which we use. In the top region there, you see the various regions on the left, some images of we also do work on modern worlds, particularly Mars and Titan and some other bodies, exoplanets and so on. And we use all the tools available to us from observational observatories. Particularly, we pioneered the use of high-resolution infrared spectroscopy for the compositional studies of comets and so on, and also pioneered the use of ALMA, the Atacama Large Millimeter Array, for planetary science. And in particular, I served on the North American Science Advisory Committee for 3 years, beginning 5 years ago, specifically to ensure that that facility could, in fact, address science of moving objects. And that was successful, and I'll show you an example of one of these key science results from that. So we look at a variety of things. And we're interested in understanding how these various processes in the protoplanetary disk and earlier in the interstellar medium could have affected the organics and water that was delivered to barren planets. The first thing you need to carry away is that although cometary nuclei now have been imaged in a number of cases, you really don't learn a whole lot from the images themselves about the origin of the material. You really have to do detailed sampling of real authentic samples, or you look at remote signatures of gases and so on in the cometary coma, for example, or in the use of mass spectroscopy, do it around nucleus of comets such as Rosetta, shown at the upper right. We do this now. In 1996, there was a paradigm shift. And the paradigm shift that occurred was, for the very first time, we were able to measure water and half a dozen organic species, nitriles included, from the ground using high-resolution infrared spectroscopy, using a new instrument at the NASA Infrared Telescope facility in Hawaii. Our team led that effort and published that work in Science. And this actually enabled our successful proposal to pursue this work in CAN 3 several years later. With red arrows here, I'm showing just the molecules now that are typically measured in a given comet at once, simultaneously. And you can see there are about a dozen different species, including water vapor. We invented the method for detecting water vapor from the ground, which we first identified on the Kuiper Airborne Observatory in Comet Halley in 1985. So this is now going on, and with help from space observations of carbon dioxide, we now can look at the chemical diversity of these bodies to some degree. Now we need more help, and James Webb is going to bring that along. So there's this future that has to do with a lot of that work from Webb and other space observatories. But the bottom line, if you look at this, is that the trace gases in a particular cometary nucleus are roughly about, something on the order of 30% relative to water vapor, water ice rather, in the nucleus. But what you see is, if you look at the gray part of the pie charts that represents carbon dioxide, and it varies dramatically amongst these three comets. So right away you see that the diversity of these comets is something we really have to get a grasp on in order to understand what the representation is for different taxonomic groups within the cometary population, and which ones of those could have been delivered to the young Earth to help deliver organics and water. Now, one of the most important things that's occurred very recently is shown in this chart. We began to realize that when you compare the two molecules, HCN and ammonia, each of those is very poorly correlated with the water production rate in the comet, but they are very strongly correlated with one another. And also they're correlated with ethane, much better than with water. And so if one looks at the ratio of for example, HCN with respect to ethane, and shows that as a function of heliocentric distance, you see suddenly that you begin to see a high enrichment in the HCN ratio at small heliocentric distances, where tiny grains, 100 atoms or so in size, can be flash heated by individual photons, such as Lyman alpha, to temperatures that permit the vaporization of the material in the grains. And in particular, if you do that again for ammonia, you can see it even more dramatically in this case, looking at the points on the right of this chart, that for comets within half an AU of the sun, the ammonia suddenly is becoming far more abundant in the coma, and it's present as an extended source, not coming directly from the nucleus, but being produced by material that flows outward. So based on this, it became clear that very likely we were seeing the dissolution of an ammonium salt, in particular ammonium cyanide. So I contacted Katrin Altveg about this. She was the PI on the Rosina instrument on the Rosetta investigation of 67P. I said, you know, do you see evidence for ammonium cyanide in your data? And she replied 2 weeks later, we don't see it. I wrote back and said, you better try again, please do. It should be there. And so she did. And then she wrote back about 3 weeks later, she said, yes, I do see it. And I said, what's different? She said, well, the difference is that I began looking at the detections of material from a solid phase particle that entered the stagnation phase of our mass spectrometer in a particular date. And I found that we were seeing these salts produced by the dissolution of tiny grains in the comet. So now here is a little chart which shows you in the green circles or ovals the particular salts that are already identified by this group. Ammonium cyanide, ammonium cyanate, a tautomer of urea, which is an organic molecule, ammonium formate, which is a source probably of formic acid in the cometary coma, seen as an extended source. And most interestingly, they also see an extended source for glycine, the simplest amino acid. And this means that it's probably being produced, stored in the comet in the form of an ammonium salt. Now, what's really interesting about that astrobiologically is it means there probably are other amino acids stored in the same way. But the bottom line is that when this material falls onto the young Earth, a comet is very weakly bound. And this material basically is delivered to the surface often intact. Let's say there's a fireball, but on the fringes you still deliver things intact. What's interesting is you deliver the water from the nucleus, you deliver the salts, and you deliver other species, but those salts will be dissolved in the water. So immediately you have a way to get the amino acids, urea, and other prebiotic molecules into the solute phase in an enriched way. You're creating this so-called primordial soup right smack in that region of the impact zone. So you don't have to worry about impacting with a rock, say a kilometer in size or so, and then letting it dissolve somehow, which is hard to do. You get it all at once in the comet. This is a very important new insight that has come out through this collaboration. Now, the second thing I want to comment is it's also very important for the case of meteoritic studies. I'm going to skip on to those. This is an image of Tagish Lake, a carbonaceous C2 meteorite. And in these, the Dworkin team has been studying the nucleobases in an array of these. It's been published back in 2011. Also with the amino acids, the LD ratios has also been studied in detail, and now about, I guess, 2 or 3 dozen objects. And finally, the amines and various others are recently published by Aponte et al. But what's really key is that the issue of the salts, thank you, is also important for these studies, because when the samples are evaluated, basically, they boil take a crushed extract and then boil that in water for 24 hours in a sealed vessel, which of course, if you have salts present, they will dissolve. And so then the question is, what is the LD ratio of the amino acid that's extracted in that case? If it were present in the salt phase, it might not even be something that is easily understood in terms of the origin of the L excess in life on Earth. Now, we have a couple of other things we've done since then I want to just briefly touch on. Mars itself, was it ever alive? Well, of course, many people in this room are aware of the paper we published on methane plumes back in 2009. This has led to 3 separate flight investigations, including Curiosity, which is now on the surface and measuring both background levels for methane and also methane spikes or plumes. Also, it's got the Trace Gas Orbiter that the Europeans have in orbit. We're involved, our team members are involved in that as co-investigators. And it has produced other information, which if you look at the lower left panel, You can see that the background level measured by curiosity is shown as the group of points at the top, right up in here. And on the other hand, the values that are returned in solar occultation by trace gas orbiter are much lower. The problem with solar occultation is it really can only sample methane down to about 10 or 20 kilometers above the surface. And the reason is that dust opacity causes extinction and reduces the signal dramatically. So we don't yet know what's going on here. But one of the possibilities is that we're seeing the important effect of interactions between the atmosphere and airborne dust. The airborne dust can, in fact, undergo saltation when it strikes the surface, be charged, and then basically cause the oxidation of methane that collides with its surface. This has been verified in the laboratory. We also have a number of other things. Water, I don't have the time to discuss. But moving on to Titan, we use now the ALMA instrument to search for the best candidate for cell membranes on Titan, which the best one is revealed by a study of the Cornell scientist led by Stevenson, was that acrylonitrile C2H5CN would be the best one. And lo and behold, if you look at this chart on the right, the top left panel shows the spatial distribution and detection of acrylonitrile by Palmer in the first paper et al. And the second paper shows the maps. I would point out to you that in our summer program for undergraduate students, Maureen Palmer was one of our brighter students in recent years, and we invited her to stay on for a 2-year gap period until she went on to graduate school, where she now is. And in that period, basically, she led the team that, well, under Martin's direction, she actually carried out the work and then published a paper in Science Advances on this initial fundamental discovery. We do the same thing in other areas, in particular advancing protocols for direct sensing on planetary missions. The MoMA instrument is now awaiting launch on ExoMars 2020. Will Brinkerhoff, our co-I, is the lead on that. And the actual protocols for that were developed under NAI funding at Goddard. Finally, the other one that's of interest is actually being able to do informational polymer analysis on places like Mars and Europa. And what's interesting about that is we really began the work with MinION technology to do DNA sequencing as a result of discussion when I was a member of a group helping the Canadians set up their astrobiology program about 5 years ago. And I got in a conversation with Deemer, and Dave told me about this MinION approach, which many of you know about, for sequencing DNA. So I brought that idea back to Goddard and triggered Jason and his postdoc, Aaron Burton, also a prior fellow from here at Ames, to begin working in the lab with that device. And that had then the following consequences. First, when Aaron left Goddard and went down to Johnson, he took the ideas with him, and is still collaborating. So he and Jason are co-Is on the first nanopore DNA sequencing, which was done in the International Space Station. It was published 2017. That work started under NAI sponsorship. And similarly, the second paper, which is Mark Sutton, who's another summer student funded by the Undergraduate Research Program Associates in Astrobiology, followed up on that, and he and Aaron, basically at Goddard, measured the radiation tolerance of the MinION technology for life detection on Mars and Europa. So I'm going to quit there because Dave's about to give me the hook.
Well, we have a bit of a fragment of a lunch session here. I guess the plan is to reconvene at 1:30. You want to shoot for that? It's above my pay grade, but anyway, thanks everybody. It was really a lot of enlightening insights.